I'm Kurt Thorn from the Nikon Imaging Center at UCSF, and today I'm going to talk about digital image analysis. And this is part one of a two-part set of lectures. And in this part, I'm going to talk primarily about what digital images are, how you display them on your computer, and how you can save them so you can work with them later. So what a digital image is, essentially, is a set of measurements of light intensity. So we have a camera. We've talked about cameras in other lectures, or a confocal microscope, or some device that records light intensity at a set of points in your field of view, in your microscope. And so you get an image like this one here, where we've got a bunch of bright objects in a field. And if you zoom in on one of these objects, you can see the individual pixels that make it up here. Some of them are bright, some of them are dark. And what each of those really represents is a measurement of the light intensity that was recorded at that point on the camera or at that point in the image. And so if you look at the numbers that make this up, you can see here this array of numbers that corresponds to the pixel intensities in each one of these pixels. And you can see that they range from zeros here at the edges where it's totally black to 200 or 255 in the center where it's very bright. And so these images are just really an array of numbers which represents the light intensity at each point in the field of view. And computers store numbers in binary formats, and in binary you can have a large number of different representations of the number depending on what resolution or what dynamic range you want. You can store it with a different number of bits. So a single bit here at the top can represent 0 or 1, so this would be a binary image. It's either black or white. If you do 2 bits, now you can have 4 colors here, this bar here, and now you've got black and white plus 2 shades of gray. And you can keep adding bits to get more and more shades of gray. So if you do 4 bits, you get 16 shades of gray. If you do 8 bits, you get 256 shades of gray. And then you can do 12 or 16, which will get you 4,000 or 65,000 shades of gray. Computers, for historical reasons, tend to store numbers as bytes, and a byte is 8 bits. So these file formats here on the top, or these bit depths on the top, can be represented as a single byte. So 1 byte will store 1 pixel's worth of information. Whereas below this line here, when you go above 256, you have more than 8 bits, you now need 2 bytes. So you would store these generally as 2 bytes or 16 bits. And so the higher the intensity resolution you want, the more dynamic range you want, the more space it takes to store your data. But also the more fine resolution you get of different grayscales in the image. Here I'm showing an image at a number of different bit depths. And at the top left corner here is the original image, which is an 8-bit image. It's got 255 grayscales. And then in these other panels, I've reduced the number of gray levels and reduced the bit depth to smaller numbers. So down here you can see what happens if we make this into a 6-bit image. And we have a fewer number of gray levels in the image, but it's actually very hard to see the difference between these two. And that's partly because your computer monitor can only show 8 bits of grayscale, and partly because your eye is not sensitive to very many shades of gray. Your eye at best can resolve 100 or maybe 150 gray levels. But then when we start reducing the bit depth further, if we go to a 4-bit image here, you can now start to see the sort of posterization where we get, especially in the smooth gradients, large discontinuities in the intensities because we don't have enough gray levels to make a smooth shading. And in 2 bits here, we see, at only 4 grayscales, we get a very stylized image that doesn't bear a lot of resemblance anymore to the original intensities here. So the more bits you have, the more gray levels you can record. And modern cameras can quite easily record more gray levels than you can resolve with your eye. And as I mentioned, your monitor, computer monitors are 8-bit displays, so they can only display 8 bits of grayscale information, so 0 to 255. They're color, so they can display 0 to 255 in each of red, green, and blue. But if you're just looking in a single color, you only get 8-bit resolution. So before, when I was showing these 16-bit grayscale traces on the bottom, we were scaling them such that the blackest pixel, the 0 pixel, was black, and the brightest pixel, the 65,000 pixel, was white. But what if instead we just scaled them so that the actual intensity values are mapped directly onto the monitor, so that 0 would be black and 255 would be white? And then you get something that looks like this. you can see the small bit depth images here are only using the very darkest gray levels on the monitor, and so they're very hard to resolve. The 8-bit one here is obviously matched to the monitor, and so we see the full gamut going from black to white. And then when we get to these larger bit depths, the 4,000 or 65,000 gray levels, you now see that all the dynamic range is compressed into this little tiny bit at the bottom, and then everything above 255 now just shows as white. So this should hopefully bring up this point that if you're looking at images that have a different bit depth than your monitor, you need to do some kind of matching of the gray levels in the image to the gray levels on your monitor. Or you're going to either compress everything to the very dark shades or saturate a tremendous amount of stuff at white, unless you happen to be working with an 8-bit image. So you always have to think about how you're going to do this mapping. And generally, this is called a lookup table in most image analysis packages. And the idea here is you have the digital numbers that make up your image on the bottom axis here, and on the side axis here, you have the display intensity on your monitor. And so here we can take an 8-bit image, and we can control how we map the intensities of the image to intensities on the display. And here we're just doing the obvious thing of mapping with a straight line that goes from 0 to 255. If instead we use a much steeper line that now goes from 0 to, say, maybe 50, and so we're saturating everything above 50 at white, you can see now the image gets very bright and washed out. So this is, for an 8-bit image, you can equally well imagine how you would do this if this bottom scale, instead of going from 0 to 255, went from 0 to 4,000 or 0 to 65,000. And from preparing images for presentations, so say putting together a PowerPoint lecture like this one, or preparing figures for a journal or publication elsewhere, almost always you need 8-bit files there, because computer screens are 8-bit and publishing workflow is designed around 8-bit files. And so if you have some image that has a larger bit depth than 8-bit, if you have this 12-bit image here, it goes from 0 to 4095, you need to think about how you're going to do this mapping. And an important point here is you lose information in this process, because we only have 255 values in our final image, but we start with 4,000 values in our initial image. And so, for instance, all the values here in this mapping, where we're just mapping from the min to the max in a straight line, all the values between 4,080 and 4,095 are going to end up mapped to 255 in the final image. And so generally this means you want to put off this conversion as long as possible. You should do this as the last step in your image analysis pipeline. If you do it too soon, you lose this information, and then that may come back to hurt you in the end, that you're trying to bring out some detail that was in the original image that then got lost when you did this conversion. So generally you want to try and stay in the native bit depth of your image as long as you can, and then as the very final step, make an 8-bit image that you can send to the publisher or put in your PowerPoint. So in this intensity scaling here, in this lookup table, you'll see a number of terms that refer to how you can do the scaling. And right now we're just talking about linear scaling, and so you can define the line as either the min and the max. That's how I've been talking about it. But you'll often also see terms of contrast and brightness. And so contrast refers to the slope of this line. So if you make this line steeper, you would get higher contrast. If you make it shallower, you'd get lower contrast. And then brightness is then the offset of this line along this plane here. So you'll see most software packages will let you set this either by setting the min and the max or by setting the contrast and brightness. And so here again is just this image and showing the effects of this brightness and contrast adjustment. And now in addition to the image, we're showing the mapping down here with a line, and then also in this gray scale here, the pixel intensity histogram. And so what this is just going through this whole image and looking at every pixel and counting up how many pixels there are at a given gray level. And so you can see in this image, there is a big peak down here of dark pixels. That corresponds to the ground. And then another big peak here of bright pixels, which corresponds to the sky. And then a smaller number of pixels of all the stuff between them. And so you can see here, in fact, that this image doesn't have anything that's completely at 255 or at 0. That the ground is not totally black and the sky is not totally white. And so we can then adjust the scaling here so that we're scaling just between the minimum value in the image and the maximum value in the image. And then you can see this makes the ground totally black and the sky in the brightest parts totally white. And so this is called brightness contrast adjustment. And this is kind of a fundamental tool you'll need to adjust anytime you're looking at an image on a screen, and particularly if you're looking at an image that has a bit depth that's different than the screen you're using to display it on. So a corollary of this is that you want to be careful how your software is scaling your image. So many software packages will scale in different ways. And the two common defaults are either to scale to the full range. So here's a 16-bit image that goes from 0 to 65,000. And the scaling here is set so that it's just mapping 0 to 0 on the monitor and 65,000 to 255 on the monitor. And so you can see this image is really dark and murky because, in fact, there aren't any pixels here that are at the maximum intensity value of the 16-bit format. There are no 65,000 valued pixels. So an alternative way to scale this is sort of how we did that image on the previous screen, which is to scale to the min and the max. And so if we do that here, now we scale between, whatever that number is and 10,000 or so, which represent the darkest and brightest pixels in the image. And now you can see you bring out this image much better, and you can see that this is an image of a galaxy and some stars. And so it's very common for software packages to do both of these things, either to default to scaling to min and max and the full range of the data, or to auto-scale to the min and max values in the particular image you're looking at. In particular, this auto-scaling, it's very nice because it makes it immediately easy to see what's in your image. But if you load two different images and auto-scale them, they're not going to have the same scaling, and so you can't directly compare intensities between them by looking at them. So it's important to be aware of what your software is doing. Another option here, of course, is that we could, instead of scaling just to the minimum and maximum values in this image, we could saturate some of the values in this image. We could instead choose to scale from the minimum to something below the maximum. And the result of that is we set all these bright pixels here to 255 on the screen and fully white, these bright pixels here to 255. And so we can't really resolve what's going on inside of those stars now. But instead, we can see very clearly this nice spiral galaxy here, which was dimmer and not really well resolved on our monitor before. Another option for scaling here is to go beyond just simple linear scaling and instead apply some kind of nonlinear scaling. And a very common one you'll see is called gamma correction. And this is basically an exponential or a logarithmic mapping of your data. And gamma equals 1 here corresponds to linear scaling. So the straight line in the middle here is the gamma equals 1 case. And then gamma values less than 1 basically saturate things at high intensity and really stretch out the intensity differences among low intensity objects. And so you can see here the slope of this curve is very steep for low intensities on the input. And so we're making a lot of grayscale distinctions here between low intensity objects. But then as we get to higher intensity objects, we're flattening those out and not using as many grayscales to represent the really bright objects. Conversely, a gamma above 1 does the opposite. So it will compress all the dim stuff here to a small number of grayscales and stretch out the grayscales among the brighter objects in the sample. And these are less commonly used, but they're very handy if you have images that have a large dynamic range where there's important differences concentrated at one end of the intensity spectrum or the other. And so just showing that applied to this galaxy image, here's gamma equals 1. Gamma is above 1. You can see here really make it hard to see the dim stuff in the object. But here, a gamma below 1 helps you bring out those dim background pixels that make up that spiral galaxy that otherwise would go missing. OK, so with all these things we can do to our images, what's acceptable? What's sort of the scientific standard here for what you can do in a published paper? And the Journal of Cell Biology has put a lot of work into this, and they have some very nice guidelines, which you can find at these websites here. But to summarize them briefly, the general idea is that brightness and contrast adjustments are OK, so long as you do them over the whole image and you don't obscure or eliminate background. So you don't want to set your 0 level so high that you suppress real background signal in your images. And you also don't want to treat different objects in your figure differently, or different objects in the same field of view differently. They require that nonlinear adjustments like gamma corrections be disclosed. And importantly, you can't cut and paste regions within an image. So you can't make a composite of multiple images where it's not obvious by white bars between the images or saying that you're making a composite. They don't want you to put things together that weren't actually together in real life. And finally, and a particularly critical point is to make any real comparisons in your image, you have to treat your control and experimental data identically. So you want to show them using the same scaling and the same gamma correction if you've done a gamma correction and so on. OK, so that's sort of the basics of how to display grayscale images. And now I want to talk a little bit about using color. So far, we've completely restricted ourselves to using black and white grayscale image displays. But there's no reason you can't use color. And so here's an example of just a 2-bit image. It's got 4 different values in it, 0, 1, 2, and 3. Here are the pixel values, and here's what this thing looks like. It's a little bright spot. And so we can display it in grayscale the way we've been doing all along right here. But we can also map different gray values to different colors on the display. And so shown here is a color map where we're using black and shades of green to display this object, and then that false colors it green. But we don't need to stop there. We can use bizarro color scales like this one, which map 0 and 3, the darkest and brightest thing, both to red. And you can see what it looks like there. You can use whatever color scale you want, basically. And that sometimes is a good way to bring out small differences in your image. So here's an example looking at kind of a fairly uniform grayscale image. This happens to be a brightness or a flat field correction for a microscope, where we're just looking at the uniform illumination on the microscope. And we've scaled this to the min and the max in the image. And so here's the grayscales. They run from 7,000-ish to 8 or 9,000 here. And you can see a lot of structure in this, but that becomes a lot clearer if we now false color this rather than just in shades of gray, and this color map, this heat map that goes from blue for dark objects all the way up to dark red for bright objects. And you can see when we do that, now we can make out fine details in this image more easily. And it also makes it easier to map the shades of color here to intensities from looking up on this color scale here, just because it's easier to distinguish differences in color than it is between 2 grayscales. We can also use this procedure to make false colored images. So here's an example of taking 3 images. These are 3 color stained tissue culture cells stained for nuclei up here, mitochondria here, and actin down here. And shown on the left here is just the grayscale mappings. And then on the right is applying the particular color scale here in these bars in the center to that image. So we've mapped the nuclei in cyan, the mitochondria in yellow, and the actin in the sort of magenta color. And if we now put all 3 of these images together, if we overlay them, we get this very pretty funky colored image. And so that brings me to color images. So most microscopy, especially most fluorescence microscopy, is done with grayscale images, just because our cameras are monochrome, and that's usually what we detect. But color images are also obviously quite important, particularly for making figures for publication, as well as for non-microscopy applications or microscopy applications where you're looking at, say, H&E stained specimens or other pathology type specimens where you want a true color image of your sample. So a color image is nothing more than 3 grayscale images, 1 image each for the red channel, the green channel, and the blue channel. And these, like any other images, can have different bit depths. More or less standard is that they're either 8 or 16 bits per channel. So if we look at just a color image here, this can be decomposed into these 3 images of red, green, and blue. And then each one of those is really just a monochrome image here with the appropriate lookup table applied to it. So a red lookup table for the red, green lookup table for the green, and a blue lookup table for the blue. And these can be stored in a number of different ways. You can either store for each pixel, the red value, the green value, the blue value, and then for the next pixel, the red value, the green value, the blue value. Or you can really just store these 3 separate images as 3 planes. A red plane, which has all the red pixels, a green plane that has all the green pixels, and a blue plane that has all the blue pixels. You don't really need to worry about this most of the time because your software should take care of the loading and properly sorting out how to display the color information in a color image. In microscopy, it's common to have more complicated image formats than just black and white or color. You often have stacks or sequences of images, say if you've taken a movie or maybe you've taken a through focus section here, a set of different z-planes in your sample, or other variables, more colors than just red, green, and blue. You could do 5 or 6 wavelengths in an image. You could do multiple stage positions, say in a 96-well plate. And so that would give you this 3-dimensional stack here where you've got a 2D image for each one of these planes. And in fact, in microscopy now, it's very common to do not just stacks, but what people call hyperstacks or multi-dimensional images, where maybe you've got a time series, and at each time point you've got z positions, and at each z position you've got multiple colors, so you've got a 4-dimensional image stack. And so how to store and handle all this data becomes a bit of an issue. And so that's what I want to talk about now, file formats and how you store and work with this data. And in particular, images can get quite big. Well, not the images themselves so much as the whole data set. So a very common camera used in microscopy has these dimensions. It's a 1392 by 1040 pixel camera, so it's about 1.4, 1.5 megapixels. And it records 14-bit images, so you'd need 2 bytes to store the data. So that requires 2.8 megabytes just to store a single image from that camera. So now imagine you're doing some complicated multi-dimensional experiment where you're acquiring 3 channels. At each position, you're acquiring 15 images, and then you're doing this whole thing for 200 time points. So at each time point, you go and you record your 15 z positions, and then at each z position, you record your 3 channels. So if you multiply this all out, you've got your 2.8 megabytes times the 3 channels times the 15 z positions times the 200 time points, and that works out to a 25 gigabyte data set. And this is not a particularly complicated microscopy experiment. You might often see this combined now with doing this in multiple positions in your sample, and this just gets bigger and bigger. So data sets that are 10, 20, or more gigabytes are not uncommon. I know people who routinely record 200 gigabyte data sets in an evening. And so obviously, if you're storing data sets this large, you need to really think about how best to store them, and particularly how to keep track of all the information of where each one of these images came from. This now has thousands of images, and you need to know the z position and time point in the channel that each image corresponds to. And this also brings up this issue of compression, because you'd like to not store 25 gigabytes if you don't have to. So there are 2 kinds of compression used for images, or used for things in general. And there's lossless compression and lossy compression. And knowing the difference between these is really critical. So lossless compression is exactly what it sounds like. It preserves all the information in your original image. It compresses your image by removing redundant information that can be restored after the fact. So this means you can always get back your original image, your original raw data from this image, despite the fact that it's been compressed and takes up less space. Most compression you're probably familiar with, however, is not lossless, but it's lossy compression. And so this is the kind of compression that's used for sort of consumer cameras for standard imaging with your iPhone or with your digital camera. And in order to make images smaller here, what this does is it throws out image data that's not visually obvious. So it removes small details in the image that you can't really see. But there is real data there that's being thrown away. And in fact, if you turn up the compression on one of these lossy compression algorithms, eventually you start to see the artifact it's introducing. But most of the time, people who write these algorithms try and design it so that the way it throws out information is not really obvious to your eye, but it is definitely throwing out information nonetheless. And that means you can't get back your raw data here. Once you've gone through one of these compression algorithms that's lossy, JPEG is a very common one, you've lost information about your image and you can't get it back. And so that means for doing scientific work, these are really terrible ideas, because you've corrupted your raw data, and you may be introducing artifactual stuff in there, introducing artifacts into your data that may affect your conclusions. And there's a really great write-up on this website here about data compression for both images and video. But the take-home message is for any scientific work, you want to be using lossless compression and not lossy compression. So I want to just talk about file formats a little bit. One of the most common file formats you'll see used in imaging is TIFF. It's a fairly old file format, but it supports both 8 and 16-bit images, which is nice because we work with both. It supports lossless compression, which is also nice because we don't want to use lossy compression. And it supports grayscale or RGB. The downside to it is that in general, TIFFs store only a single image or a single color image. And so that means in our example where we're taking 3 channels and 15 Z-stacks and 200 time points, we're going to need thousands of TIFFs to keep track of all those images. And so that's not so nice. There are other file formats. There's a new variant on JPEG, JPEG 2000, which you don't see that much, which is lossless, which is quite nice. And then basically to deal with this issue of keeping track of these thousands of images, most of the microscopy companies have come up with their own custom formats. So you see things like ND2, that's a Nikon format, ZVI and LSM are Zeiss formats, and so on. And these are generally pretty nice. They're lossless. They support the full bit depth of your system. Almost always, those custom formats will support multi-dimensional images. So you can save that entire 3-channel by 15 time or by 15 Z-slice by 200 time point data set as a single file, a single large file, but a single file. And these custom formats will also often keep track of the metadata about your experiment. They'll keep track of things of like how your microscope was set up, what the Z spacing was in your Z stack, what the different channels you used were, what the time interval in your time lapse was. And so those are a very nice option, but the downside to them is because they're custom, they're not always so portable. So you certainly won't be able to open these in something like Photoshop. You can always open them in the manufacturer's software, and generally you can find plugins to open them in things like ImageJ. But if you want to get these into, say, a custom program for doing image analysis, you'll probably have to convert them to a TIFF stack first. So a whole bunch of TIFFs and then load those into your program. And then there's a whole bunch of file formats you probably want to avoid. And these are things like JPEG, as I said, it's lossy. This old GIF format or bitmaps and all these formats that were designed more for ease of use or computer simplicity than for really preserving data. So many of these are restricted to 8-bit only, and some like JPEG also use lossy compression automatically. And so in general, they require corrupting your data in some way you probably don't want to do. They may be fine for putting pictures on your website, but you don't want to use them in your scientific data processing chain. So finally, I want to just say a few words about the software tools that are out there for doing image analysis and working with images. There's a really large number of them. So I'm not going to go into any detail here, but just to sort of summarize what's out there. So there are a large number of programs that will do both acquisition, meaning they'll control your microscope, and do data analysis. And so there's a large number of commercial ones. NIS Elements is Nikon's software. Axiovision is Zeiss's software. Metamorph is a software package that's been around a long time that's microscope independent and will control many different microscopes. Slidebook is a similar program to Metamorph in that way. And then Micromanager is one I want to touch on because it's developed at UCSF and it's free and open source. And it's basically a plugin that lives inside of ImageJ and will control microscopes. And then you can use the ImageJ functions for data analysis. So all these programs will let you both control your microscope and do many different data analysis tasks. Then there's a bunch of presentation tools, things like Photoshop, which you're probably familiar with, or the freeware version of this, the open source version, GIMP, which are designed not for really doing scientific data analysis, but for making attractive figures and putting data together so you can display it in a figure or in a presentation. And then there's a bunch of sort of dedicated image analysis packages, and these include things like MATLAB, which is really a general purpose programming language in many ways, but it has very nice tools for doing image analysis. ImageJ, which is this open source free tool that has many different image analysis routines built into it that you can use for doing image analysis. Imaris, which is a commercial package, which is really optimized for doing high-end 3D visualization. Cell Profile, which is another free open source tool that's optimized for doing high throughput screening analysis, so in analyzing thousands or tens of thousands of images automatically. And there's many others I haven't mentioned here, which are sometimes specialized for particular tasks or just a little less commonly used than these. So that's all I'm going to say today, and I want to just thank Nico Sturman, who provided a lot of the slides and the outline for this talk. Thank you.